CRISPR-Cas9 is a versatile genome editing platform derived from an adaptive immune system found in bacteria and archaea. In its natural context, CRISPR loci store fragments of viral DNA that, together with CRISPR-associated Cas proteins, enable targeted recognition and destruction of invading genetic material. In 2012 to 2013, this system was adapted for programmable DNA editing in eukaryotic cells, transforming biological research and opening prospects for therapeutic and agricultural applications. At its core, the CRISPR-Cas9 system requires three conceptual elements: a DNA targeting guide and a nuclease that makes a cut. A short RNA sequence, guide RNA, is programmed to match a chosen DNA site. The Cas9 protein binds the guide and scans the genome until it finds a complementary sequence adjacent to a short recognition motif called the protospacer adjacent motif (PAM). Cas9 then creates a double-strand break at the targeted site. Cellular DNA repair pathways act on that break, and by influencing those pathways, researchers can disrupt genes, introduce small insertions or deletions, or, under appropriate conditions, install new sequences. Key features and variants: SP-Cas9 and other orthologs. Different Cas9 proteins recognize different PAMs and have varying sizes and properties, enabling flexibility across systems. Single guide RNA, sgRNA, a synthetic fusion of two natural RNA components that simplifies targeting to a single programmable RNA molecule. dCas9, dead or nuclease deactivated Cas9. Catalytically inactivated Cas9 can be repurposed as a DNA binding scaffold to modulate transcription, visualize loci, or recruit epigenetic modifiers without cutting DNA. Base editors and prime editors, engineered derivatives that enable precise single base changes or short sequence edits without creating a conventional double-strand break, expanding the range and precision of edits. Major applications: basic research, rapid generation of gene knockouts, knock-ins, mutations, and pooled genetic screens across many organisms. Therapeutics, development of somatic cell gene therapies for monogenic diseases, cancer immunotherapies, and ex vivo engineering of patient cells, e.g., edited T cells or hematopoietic stem cells. Agriculture and biotechnology, trait improvement, disease resistance, and functional studies in crops and livestock. Diagnostics, CRISPR-based detection platforms that exploit collateral nuclease activity for sensitive nucleic acid sensing. Functional genomics and synthetic biology, multiplexed editing for pathway engineering and large-scale functional studies. Advantages: programmability. Targeting is directed by an easily synthesized RNA sequence rather than requiring custom proteins. Scalability and multiplexing. Multiple sites can be targeted simultaneously. Broad applicability, works in a wide range of cell types and organisms. Limitations and risks: off-target activity. Imperfect guide-target pairing can lead to unintended edits elsewhere in the genome. Assessing and minimizing off-target effects is a major focus of ongoing research. Repair outcomes: cellular repair pathways can be unpredictable, producing heterogeneous outcomes, e.g., insertions, deletions, or mosaicism in multicellular organisms. Delivery challenges: efficient and tissue-specific delivery of the editing components in vivo remains a central technical barrier. Immunogenicity, pre-existing immunity to bacterial Cas proteins and immune reactions to delivery vectors are potential safety concerns. Ethical and societal considerations: germline editing raises profound ethical questions because changes are heritable. Equitable access, consent, and long-term monitoring are important regulatory and social issues. Regulation, ethics, and governance: regulatory frameworks differ by country and evolve as the field matures. Somatic cell therapies undergo rigorous preclinical and clinical evaluation for safety and efficacy. Germline editing for reproduction is widely discouraged or restricted by international consensus and national regulations. Responsible development emphasizes transparent oversight, stakeholder engagement, and adherence to ethical norms. Future directions: improvements in specificity and control, novel Cas variants and smaller editors, advanced delivery vehicles, and refined editing modalities, such as base and prime editing, are active research areas. Advances aim to expand the therapeutic window, reduce unintended effects, and enable precise, durable interventions in vivo. As technical capabilities advance, parallel progress in ethical frameworks, governance, and equitable access will be essential to realize societal benefits while minimizing risks. Overall, CRISPR-Cas9 has reshaped genetics by providing a powerful, adaptable toolkit for manipulating genomes. Its continued development promises substantial scientific and medical advances, tempered by technical challenges and important ethical responsibilities.